For a long time now, we've gotten really good at reading the code of life. But what if we could actually write it? I'm not talking about copying it, but creating brand new DNA from scratch, just using chemicals in a lab. Let's get into the incredible story of how we learn to write the very instructions for life.
I mean, that sounds like something straight out of a movie, right? But it's the real question at the heart of synthetic biology. The dream of designing organisms that can create medicines or biofuels or new materials. It's happening. And it all begins with one single fundamental skill. And that's the real kicker here. We're not talking about just making copies of existing DNA like PCR does. Chemical synthesis is building DNA from the ground up, letter by letter, without any template at all. It's pure creation. But man, figuring out how to pull that off was a massive challenge. So to make this dream a reality, scientists first had to solve a really tricky puzzle. How do you get these tiny DNA building blocks, the A's, T's, C's, and G's, to connect in the exact order you want without them just clumping together in a random mess? The answer, it turns out, was a brilliantly simple idea.
Think of it like this. If you want to write the word cat, you need to add the C, then the A, then the T, in that order. Protecting groups are like putting a temporary cap on the top and bottom of the letter A. That way it can only connect to the C on one side and wait for the T on the other. It's all about forcing the chemistry to happen in a nice, orderly line.
Okay, so with this idea of protecting groups in hand, the first real attempts got started. But you know how it is with brand new technology. The first drafts were, full of typos. This was one of the first big tries, the phosphodiester method. And just look at it. The problem was that central phosphate group, the little connector piece. It was left totally exposed, and it started making connections where it shouldn't, creating all these messy, unwanted branches off the main DNA chain. So instead of a clean sentence, you got total gibberish. The fix was the phosphotriester method you see on the right, and the breakthrough was so simple, yet so brilliant. They just added another protecting group to that trouble-making phosphate. That one little change stopped all the branching, cleaned up the reaction, and gave scientists their first truly reliable way to build short DNA strands. Problem solved, right? Well, not so fast. The phosphotriester method worked, yeah, but it was painfully slow and pretty inefficient. Trying to build anything longer than just a few letters was a huge headache. The whole process needed a major speed boost. And let's get real about why it wasn't cutting it. A 90-minute cycle to add one letter? That's an eternity. And 95% efficiency sounds good, but it's a trap. With every step, you lose 5% of your correct product. After just 20 letters, you're making less than 36% of what you actually want. A much faster, much more accurate method was desperately needed. The answer came from a totally different way of thinking about the chemistry. You see, the phosphorus in our natural DNA is super stable, which is great for, life, but it's terrible for speed. So scientists had this wild idea. What if they used a really high-energy, super reactive version of phosphorus for just a split second to make the connection? It was like swapping out a regular engine for a rocket engine. So you take this new, souped-up chemistry, combine it with a few other key innovations, and suddenly you have the groundwork for the technology that powers everything today, the modern automated DNA synthesizer. And here it is, the hero of the story, the phosphoramidite. Now I know this diagram looks complicated, but the idea behind it is pure genius. This is the prepackaged building block, the ink cartridge for the DNA printer. It has that super reactive phosphorus, but it's kept stable and safe until the exact right moment. All the protecting groups are already in place. It's just ready to go. The other game changer was a technique called solid phase synthesis. Basically, you attach the very first DNA letter to a tiny glass bead. The growing DNA chain is anchored, held perfectly still, while all the different chemicals just wash over it in sequence. It's like clamping the paper into a typewriter so the keys can strike one by one, perfectly. This is what made the whole thing automatable.
And this is what that automated process looks like today. It's just this clean, elegant four-step cycle that repeats over and over and over, adding one letter at a time with incredible accuracy. With every turn of this wheel, the DNA strand gets one base longer. Let's just walk through it real quick. First, deprotection. A chemical snips off the protective cap from the end of the chain, opening it up for the next letter. Second, coupling. You add the next phosphoramidite, and that rocket fuel chemistry instantly forms the new bond. Third, capping, and this is a clever quality control step. Any chains that failed to grow are permanently blocked off so they can't cause errors later. And finally, oxidation. That new, unstable bond is converted into the stable, natural form found in DNA. Then, the cycle just starts all over again for the next letter.
So this automated cycle is amazing, but it does have a limit. It can only reliably build DNA strands, or oligos, up to about 150, maybe 200 letters. But entire genes are thousands of letters long, and genomes are millions. So how in the world do we get from these short fragments to the really big stuff?
The answer is assembly. Scientists design and synthesize a whole bunch of short, overlapping pieces of the final sequence they want, and then they cleverly use the cell's own molecular tools to stitch all those little pieces together in the right order.
And there are two main ways they do this, both borrowed right from nature's playbook. The method on the left uses an enzyme called ligase, which is basically molecular superglue. It finds the perfectly matched ends of two fragments and just bonds them together. The method on the right is more like weaving. It uses polymerase, the same enzyme from PCR, to fill in the gaps, using the overlapping ends as a guide to weave all the short pieces into one long, seamless DNA strand.
And the scale of what we can now build is just staggering. We went from a 77 base gene in the 60s to the first 7,500 base viral genome in 2002 to today where scientists are writing entire bacterial genomes. We're talking over 500,000 bases, completely from scratch. The progress has been absolutely exponential. At the end of the day, this isn't just a cool chemistry trick. The ability to write DNA is the absolute foundation of synthetic biology. It is the essential tool that allows us to test our understanding of genes, design new biological functions, and engineer life in ways that were pure science fiction just a few decades ago.
So we've gone from painstakingly connecting a few chemical letters to designing entire genomes on a computer and basically hitting print. We now hold the pen that can write the book of life. The power here is just immense, and that leaves us with a profound and incredibly exciting question. What should we create next?